Hello, Anatomy students. Today we're talking about the electrical nature of neurons. You should have a pencil, you should have your lecture worksheet, and let's get started. All right, so before we start, there are some things that you need to keep in mind. And the first is that neurons conduct nerve impulses, and they are characterized as being excitable and conductive. Also, remember, we've got to talk a little bit about chemistry here, which is one of my favorite subjects. But just remember that opposite charges attract while like charges repel. And it's also important to realize this because the two main ions that are going to play a role in the workings of a neuron are both cations. They're both positively charged, both sodium and potassium. So sodium is Na with a +1 charge, while potassium is K with a +1 charge. Also remembers that ions try to achieve equilibrium, and if they were allowed to, they would just move until they reach certain concentrations across a barrier, but they are stopped by channels and other factors in the cell membranes of neurons, so that's not always going to be possible for our little ions. All right, so a membrane potential is simply the difference in charge between the intracellular fluid or the inside environment of a neuron and the extracellular fluid or the outside environment of the neuron. What this means is that the membrane is then polarized because you've got more positive charge on the outside and more negative charge on the inside. And so what you're looking at our picture on this slide is all the blue part is the extracellular fluid, and the inside of our cell, like this is like a cross section of an axon, and so the inside is going to be more negative, and the plasma membrane is there at that blue line that is circling the yellow circle in the middle. It's interesting to note that whenever you see a charge for a membrane potential, what it's actually telling you is the measurement, like the negative sign is telling you that the inside of the cell is negative, not the outside. So if you take a look at the voltmeter that you have there, you'll see that I have the -70 boxed in yellow, and that means that the inside of the cell is negatively charged, and that the difference between the charge of the outside of the cell and the inside is 70 millivolts. And so that's what that means. And like the two wires that are coming off of the voltmeter, they have electrodes that are at the end, and you can see that one is actually measuring the charge on the outside of the cell while the other one is measuring the charge on the inside. And this is definitely something that was done, decades ago when scientists were doing research. They would use the giant squid axon because it's one really long cell, and so it was very easy to insert the electrons or the electrodes in order to measure the membrane potential. And then they would stimulate it, and they would take measurements on the voltmeter, and that's how we obtained a lot of the information that we know today about how neurons work. Okay, so RMP, this is the abbreviation that we're going to use for resting membrane potential. And the resting membrane potential for a neuron is -70 millivolts. It is really important that at rest, an ionic imbalance is maintained in our neuron, because when a stimulus comes to the neuron, we want the response to be immediate. We don't want there to be any sluggishness in the response, otherwise we are not going to be able to react appropriately. So when a response comes, we have ions switching places automatically because they're set up, they are imbalanced already, and they want to become more balanced. So they're just going to do this automatically without any kind of, help from anywhere else. It just kind of happens. So the ionic imbalance is going to be maintained by four things. So on your sheet, you have A, B, C, and D. Letter A is gated channels. We have a couple of different ones. We have ligand gated channels and we have voltage gated channels. Ligand gated channels are channels where a certain molecule or a ligand has to actually bind to that channel in order to get it to open. In this case, what we're talking about is a neurotransmitter. So when a neurotransmitter binds to a neuron, it binds to a ligand-gated channel, and then it causes it to open up. Some of these gated channels are voltage-gated channels, where they don't respond to a molecule binding to it, but they respond to a change in the potential and the charge around that channel. And so if the voltage or if the membrane potential around that channel reaches a certain point, a threshold, remember that word, we're going to come back to it, then it will open up. And these gated channels are specific for potassium and specific for sodium. So, a sodium channel can't let a potassium channel in or vice versa. Letter B are leak channels, and these are channels that are going to let a trickle of potassium leave and enter the cell. And so this is also going to help keep the inside of our cell negative, because potassium is a positively charged ion, and so if it leaves a certain area, that means that that area becomes more negative. Letter C is the sodium-potassium pump, and this is an ATP-driven process where ATP has to bind to a protein in the cell membrane, and the protein is going to pump 3 sodium out of the cell and 2 potassium back into the cell. Now, why does this require ATP? Because it is pushing these ions against their gradient. Remember that ions want to flow from an area of high concentration to an area of low concentration, but on the outside of the cell, we have a lot more sodium than we do potassium. So to get the sodium from the inside to the outside, you're going to have to use a little bit of energy in order to push it out. And the same thing with potassium. We have a lot more potassium inside the cell than outside, so we have to use some energy in order to pull that potassium into the cell. And then finally, we have large negatively charged protein molecules that live inside of the cell in the intracellular fluid there. And so that's also going to help to keep that negative membrane potential. So it's important to just realize that at rest, a neuron is not excited, but it's not stable either. I know that one thing that we talk about, like when a neuron is at rest, we oftentimes will call it a salty banana, with sodium on the outside, potassium on the inside, and, we say, oh, it's resting, but it's not stable. Resting and stable don't mean the same thing. It's polarized. Polarization simply means that you have a separation of charge. And so when you're talking about the neuron, we have positive charge on the outside, we have negative charge on the inside. And so the cell is polarized and it's ready for those ions to switch, generating a potential down the rest of the cell membrane. And again, we want that to happen very quickly. Otherwise, we are not, as organisms, we won't be able to respond appropriately. Okay, so the first thing that I wanted to talk about are local or graded potentials, because these are the first things that have to happen inside of the cell. Once we get local potentials traveling to a certain point in the cell, they're going to combine, and then if they reach threshold, then we get an action potential down the axon. But, traveling through the dendrites in the cell body, these are called local potentials or graded potentials, and then all of these come together and summate, and if threshold is reached, that's when we get the action potential. So I just want to make sure that you understand right off the bat that graded or local potentials are not the same thing as action potentials. They're generated in similar ways. Let's talk about these local potentials or these graded potentials. So these are simply fluctuations above or below resting membrane potential. So when our cell becomes excited, what happens is that a neurotransmitter, a ligand, is going to bind to a sodium gated channel, and then the sodium channel opens up and allows sodium, which is positively charged, to enter into the cell. And that's going to cause a little positive blip on our voltmeter. And so if you take a look at the voltmeter picture, you'll see that you have like two bumps on the left side of the voltmeter. And these are local depolarizations. These are membrane potential changes that were positive because these the ligand-gated channel opened, let some sodium in, and then because that area, that local area, became a little bit more positive, voltage-gated channels opened up and allowed some more sodium to come in, allowing for that membrane potential to become more positive a little bit on down the membrane. So this is called depolarization. Remember what I said, that a resting neuron is polarized? Well, we are depolarizing. We're taking away the polarization when we open up the channels and we allow the positive things to come into the cell. We're reversing that separation. And I just wanted to mention, though, that sometimes it's not a neurotransmitter that causes excitation that binds to the ligand-gated channels. Sometimes it's a neurotransmitter that causes hyperpolarization. So if a neurotransmitter binds to a ligand-gated channel on the surface of our neuron, if that channel opens and allows potassium to leave the cell, then we get something called hyperpolarization, because now instead of that membrane around the area becoming more positive, it's going to become more negative because the positive ion potassium is leaving the cell, causing hyperpolarization. So that's going to look like a dip in our voltmeter reading, and that's what you see to the right of the picture of the voltmeter here. Okay, so as I said, local potentials can either cause fluctuations in membrane potentials either above if it's an excitation or below if it's an inhibition membrane potential. And so this slide here is just showing you what I was explaining before about when a ligand-gated channel opens and allows potassium to leave. Potassium goes out of the cell, the membrane potential is going to dip below 0 millivolts, and this is something called hyperpolarization. And again, this is a local hyperpolarization. This isn't the whole cell, it's just a little portion of the cell membrane that is becoming hyperpolarized. So as I said, local and graded potentials are different from action potentials. They are isolated. They're only in a particular area. They don't spread all the way down the axon. Not yet. Not yet. They can be large or small. They are not all or none events, unlike an action potential, which is an all or none event, which we'll talk about. And they're also going to exhibit something called decremental conduction, which means that as they spread down the cell body and down towards the axon hillock, they get weaker in strength, and that's not something that happens with an action potential either. So as all of these graded potentials, both inhibition action potentials and excitation action potentials alike, if they're coming in at about the same time, they're all going to travel down the cell body to the axon hillock, and they're going to combine and summate. And if they reach threshold, then they will generate an action potential. If they don't reach threshold once they add all together, well, then an action potential will not be generated in that axon. Okay, I'm going to stop this video here, and I'll do part 2 talking about action potentials. So just remember, what we've talked about so far is that graded potentials can be either inhibitory or excitatory, and they all travel down the dendrite, down the cell body to the axon hillock, and they add together. And if they reach threshold, then they generate an action potential. And if they don't, then the action potential is kind of dead in the water. It's never going to happen. All right, we'll talk about action potentials next. See you in a minute. Oh, stop.